So when we talk about personalized medicine, it really is personalized. We're talking about really understanding the genetic makeup of that patient, understanding the specific type of cancer tumor that patient has, and then targeting that cancer through immunotherapy, which is, again, very, very personalized.
Well, it's slightly different from the personalized concept of genomic medicine. It's more personalized in the fact that we take a patient's T cells that's able to recognize all those changes. We don't know what those changes are. We don't know what those mutations are, but it's the T cell's job to know what they are. So what we do is basically unleash the T cell to be able to kill the tumor cell. So the T cell is able to recognize, dozens, hundreds of mutations within the tumor cell, and able to, through that recognition, get activated and kill the tumor cell. So it's personalized in that we're taking the patient's T cells and activating it against their tumor cell, but the T cell knows everything. We don't know its genomics, but the T cell does.
Yeah. So in prior courses of therapy, chemotherapy, for example, you're injecting a patient with chemotherapy, and it's going after the tumor, but it's also destroying some of the good cells that are housed around that tumor. And that was one of the side effects or risks of chemotherapy. And in immunotherapy, we're kind of getting around that because the body is really working overtime to help fight that cancer and really hone in and target that tumor.
It's a form of targeted therapy. So these T cells recognize a tumor cell versus a normal cell with exquisite sensitivity because they have to. If when that recognition breaks down, the T cells can actually attack the body. So there's a very extensive regulatory system to make sure the T cell recognizes only bad cells and not normal cells. Mm-hmm. There are side effects and toxicities that we've been able to encounter with these therapies, but it's somewhat of a kind of a positive to these side effects. These side effects are usually related to the T cells killing the actual tumor, as opposed to just a non-specific side effect of normal cells being killed.
Got it. So tell me what a day in the life of you looks like. You go into the Cancer Center and Research Institute, and what does your day consist of?
So I wear multiple hats at H. Lee Moffitt. I am a physician scientist, so I'm an MD PhD. I have clinical responsibilities. I take care of patients with hematologic malignancies and sometimes solid tumor malignancies and try to provide them standard of care. Another hat that I wear is a laboratory-based physician scientist. So I spend a lot of time at the bench. I myself don't work at the bench, but I manage a team of scientists at the bench devising experiments and trying to ask important questions within very controlled settings of how can we make these therapies better? How can we develop new immunotherapies? And the other hat that I wear is kind of a translational researcher who's focused on taking those basic science discoveries at the bench and translating them to patients and doing phase 1 clinical trials so that we can demonstrate its safety, but also hopefully identify some potential efficacies that we can then later on develop into further phase 2 and phase 3 clinical trials. So I wear multiple hats in a day. I'm dealing with laboratory issues, I'm dealing with clinical issues. I also am the medical director of the cell therapy facility, so I have to evaluate the cells that patients receive as part of their standard of care for stem cell therapies, but also as experimental cell therapies. So it's a very, every day is different. The morning can be very different from the afternoon, but to me, it's just tremendously rewarding because I really have my hands in all these processes, going from the very beginning of a basic science discovery and bringing it to patients. So that's my motivation in terms of why I work in the lab, is because I see the standard of care is really lacking for many of these patients, and it helps me devise good experiments, and it motivates our lab staff to work hard and do great work.